Welcome to Chapter 5, and in this chapter we're going to discuss chemical reaction. So we're going to start by discussing thermodynamics, which is just the study of the energy of reactions. We're going to follow that by a study of kinetics, which is a study of how fast reactions occur and what we can do to influence the speed of reactions. So we can speed reactions up or slow reactions down, depending on what we're trying to do. Remember, in a biological context, this might be speeding up or slowing down the rate at which a molecule is synthesized or broken down to affect a disease state. So we might use a medicine to do that, but there are other ways in a flask that we can manipulate things as well. The next thing we're going to discuss is an overview of chemical reactions. So we'll talk about how chemists represent chemical reactions. So we'll talk about how we put out how we write down the reactants, and we usually have an arrow and then the products. And we'll show how that's done in standard chemistry as well as in biological chemistry. We have some different ways of drawing chemical reactions. We'll talk about some specific types of chemical reactions, particularly oxidation and reduction reactions. A lot of the way that our bodies produce and store energy are these oxidation and reduction reactions. So they're very important in the field of medicine. We'll also discuss organic reactions such as condensation and hydrolysis. These are often employed by enzymes in our body for the synthesis and breakdown of organic molecules, and they have other applications as well. And finally, we'll discuss the organic addition reactions to alkenes as well. So we're going to start off with two definitions. Reactions can be either exothermic reactions or endothermic reactions. And so all reactions can be split up into one of these two. So an exothermic reaction is an example of a reaction that gives off heat. These exothermic reactions are typically going to be reactions that continue on going as long as you have reactants to let this keep going. So these are reactions that go on their own. Usually, sometimes they'll require a little spark, and we'll talk about that in a second to get rolling. The other type of reaction is an endothermic reaction. So an endothermic reaction requires heat to go. So the difference between the two is your exothermic gives off heat. This is an energy generating process, and endothermic requires heat. So it requires work in order to build something up, perhaps. So really, the way I think about this is if you think of skeletons, if you want to try to remember these definitions, the types of skeletons that you probably learned about in grade school, high school, middle school were exoskeletons and endoskeletons. So things like our bugs, like ants, have an exoskeleton, which is on the outside. So I think of exothermic. Thermic means heat. Exo means outside. In an exothermic process, the heat is given off to the outside. So the heat is radiated out or given off. In an endothermic reaction, an endoskeleton would be inside of you. So the heat is being taken into the process. So endothermic is the heat is being taken in. It requires heat to go. So heat's a form of energy, and energy can exist in several other forms as well. So one form is randomness, how random a system is. As you increase the heat of something, you're increasing its randomness. So the molecules involved are going to bounce around more. So randomness is one thing that we need to take into consideration when we're determining the free energy of a reaction. Is a reaction going to continue on its own, or is it going to require energy to keep it going? If it increases randomness, the world tends toward chaos, or the universe tends toward chaos. So if randomness is increased, this is typically going to be a spontaneous process, or one of our exothermic processes. If you're ordering something, that typically requires energy to put something in place. Think about it as like if you're trying to, if you have a messy person, he doesn't spend much time or energy, and the room around him or her just becomes completely randomized. It becomes a total mess. Whereas if you want to order things, that requires energy. So if you're increasing randomness, typically that's an exothermic process. If you're decreasing randomness, that's typically an endothermic process. Another way that we can represent heat or energy in a reaction is just by what we call delta G, or the Gibbs free energy. This is typically measured in calories, especially in biological chemistry. So it's just a measurement of energy. We mentioned that calories are a measurement of energy that we use in chemistry, biochemistry, and nutrition. Um, so the free energy, or delta G, as we're going to describe it in a minute, is going to be the change in energy from the reactants, what you start with, to the products. So we can have energy stored in a molecule. For example, if you have a fat molecule, we know that fats are a way that our body and other organisms in nature store energy. So they used an endothermic process that required energy to build that fat. So if we take that fat and burn that fat, there's going to be an energy change. Energy is going to be given off in this process. That would be an exothermic process, the process of burning fat. So there would be a change in that free energy, or delta G, that would give off energy into our body that we can then use to move, to run, to play, to learn, to study, to do whatever it is that we need to do. So we talked about endothermic and exothermic a second ago, and exothermic and endothermic specifically just talk about the form of energy that we call heat. And so there's another definition here which is very similar, and it's what we call exergonic. And that just means gives off energy, and endergonic, which requires energy. So you think the gonic just stands for energy. So delta G includes both heat and that randomness factor that we discussed a second ago. So you could have a reaction that doesn't give off heat necessarily, but it does increase the randomness. And so we'd call this an exergonic reaction. So the exergonic and exothermic reactions, those are typically going to be our spontaneous processes. These are processes which continue to occur once started, and they don't require energy from the surroundings to allow them to continue to occur. So if you have a spontaneous reaction or an exergonic reaction, the free energy of the reactants, what you're starting with, is going to be greater than the free energy of the products. I'll show you a diagram that kind of helps visualize that here in just a second. So a real life example is we were just discussing burning fat. So if you take fat, there is stored energy in that fat. So if we're burning fat, we're going to be taking fat and using oxygen to burn that fat. Our bodies do that constantly. And so it's sort of like a combustion reaction. So we're taking that fat and we're burning it with oxygen, and we produce CO2 in the process. So the free energy of the reactants, or the stored energy in that fat, is greater than the free energy of the products. So our product, when we burn fat, we react fat, which is essentially a hydrocarbon, with oxygen. We produce CO2 and water from that. So the free energy of the fat is a lot higher than the free energy of the products. So our delta G, which is the difference between the starting energy and the final energy, is going to give us a negative value. This would not require an energy input to occur spontaneously. Once you get that fat burning, it's going to continue to burn. So an example would be if you have like a grease fire on your stove, as long as there's fat and oxygen present, that grease fire is going to continue to occur. You're going to continue to give off energy. So it's a negative delta G value because energy is being given off to the surroundings. The starting material is losing energy in this process. So that'll continue to happen until essentially you're out of reactants. So one important thing to point out is that delta G, it can predict if this is going to be a spontaneous process, but it does not imply that this is going to be a quick process. So it doesn't tell us how fast this particular reaction is. So another great example of that is the nuclear breakdown of carbon-14, which we use for radio labeling, and that particular isotope of carbon breaks down very, very slowly, although it does have a negative delta G. So it occurs spontaneously, but it occurs very slowly. So if your delta G is positive, that means that the products have gained energy in this process. You had to put energy into the system. So if your delta G is positive, you have an endogonic reaction, not an exergonic reaction. So if you have positive delta G, we can think of it kind of like you have to put energy into the system to get it to work. And we can use this example of a canoe. So a canoe can only go upstream if the paddler puts in energy. So that'd be a great example of a non-spontaneous process. If you're not putting in energy, you're just going to go backwards. You have to put the energy in order to create your new thing that you're trying to create, or to paddle against the current. So this might be one way I could represent this on a test and expect you to tell me what's going on here. So here we have in figure A, we have the G, or Gibbs free energy of the reactants, and we have the Gibbs free energy of the products. As you can see, the free energy of the products is less than the energy of the reactants. We calculate delta G by taking the energy of the products minus the energy of the reactants. So if your reactants start out with a lot more energy, you end up with a negative delta G. So energy has been given off to the system, or we've lost energy in this process. And that's why we have a negative delta G. And that's why we do the order G of the products minus G of the reactants. So we can show if you'll get a negative number, means energy was given off, or we lost energy in this process. That energy is typically going to be used for something else, like to power your body, or to build something else in biological systems. So if your Gibbs free energy of your products is lower, I would expect you to be able to tell me that A is an exergonic reaction. So energy has been given off in this process, and this is probably a spontaneous reaction. Whereas B, you would look and see that our reactants energy is lower than the energy of our products. So in this case, we're storing energy. Energy is required to make this happen. A perfect example of this is we take the exergonic reaction in A would be us breaking down a fat or sugar into CO2 and water. That would be our exergonic reaction. We're taking something that was stored energy and using it. And our endergonic reaction would be photosynthesis. So a plant is taking light energy from the sun, that's where it's getting the energy, and it's taking CO2 and water, and it's producing sugar and oxygen, both of which have more stored energy. So the products have a higher stored energy than the reactants did. So it's taking a low energy reactant like CO2, and it's turning it into a high energy product like glucose that we could then turn around and eat and repeat this cycle. So one of the best applications, or biochemical applications, I can think of for thermodynamics is the coupling of a high energy reaction to make other things happen. So in living systems, we can take the free energy that's given off in a chemical reaction and couple it to another reaction to make something happen. An example of that is we use the molecule adenosine triphosphate. This is a very high energy molecule. It's the energy storage molecule of the body. Our bodies break down sugar and we store that energy as ATP. It's made in that process. And then we use ATP to do a ton of different things in our body. So an enzyme will grab a hold of ATP, break off a phosphate. That energy is used for that enzyme to move or to create a change in another molecule or do something else. So we can take that energy from one process and convert it into energy used to drive another process that would have been an endogonic process. So here we've got two other diagrams, and these are a little bit more complicated than the last diagrams we were looking at. The last diagram just had the energy of the reactants and the energy of the products. And here we have this new energy, and it's called the activation energy. So think of it this way. If we're burning gasoline, does gasoline just burn if you leave it out? No. You've got to spark it. You've got to light it somehow. If you take a butane lighter, like a cigarette lighter or something along those lines, hopefully nobody's smoking in this class, but if you need to like light a candle or something along those lines, and you're using a butane lighter to light a birthday candle, that thing won't just light if you release the butane to the oxygen. The butane and the oxygen don't spontaneously react until you give them a little spark. So we need that spark to have this happen. That spark is the activation energy. So it takes a little energy to get these processes going, these exergonic processes going sometimes. Sometimes they are spontaneous. There are dangerous chemicals that will spontaneously react when exposed to oxygen. They're very dangerous. They're used in weapons quite often. Sometimes they're used in synthetic reactions and things along those lines. But a lot of exergonic reactions require a little bit of energy to get started. And the energy that keeps them going is the energy that's given off. So that's the energy difference between the energy of the reactants and the energy of the products is the energy that's used to keep the reaction going until you run out of reactants. So that's why you can just spark a fire. Just a little spark is required to get that fire going. So you require a tiny bit of activation energy to get this process to go. But after that, the energy that's given off by the products is going to be what drives the reaction. So this is your spark, your activation energy, and this is the energy, the free energy change of that delta G. So this is an example for exergonic reactions. In an endergonic reaction, you're going to have a much higher activation energy. And then the free energy change is shown here. And that's, again, the difference between the energy of the reactants and the energy of the products. So endergonic reactions require a lot more activation energy typically, because their activation energy is always going to be, I shouldn't say they require a lot more activation energy. That's incorrect. So their activation energy is going to be higher than their free energy change. So takeaway, what you would need to know on a test if I show you a diagram like this, you'd have to be able to identify what is the activation energy, what's the energy of the reactants, the energy of the products, and what's the free energy change. So the free energy change is the difference between the energy of the reactants and the energy of the products. The activation energy is the difference between the energy of the reactants and the top of the peak here that's shown in both of these figures. I won't have anything about calorimetry on the test, but this is kind of cool. This is one of the ways that we measure the amount of energy that is present in foods. If you ever wonder how they get those calorie counts on the sides of a box, it'd be really hard to go into the human body and measure exactly how much energy is given off by, a bit of food that you eat, because we often break it down and store it. We're not constantly burning what we're putting into our bodies rapidly. So we have a device called a calorimeter, and a calorimeter burns the food in the presence of oxygen, just as our bodies would do. Our bodies use a more complex series of chemical reactions, but the amount of energy given off is pretty darn close to the same. So this is one easy way that we can measure how much energy is present in food. So the food's placed inside a closed container, is surrounded by water and burned, and the temperature change is measured, and that's going to directly relate to the amount of energy that was released from this product. So this will give us a measurement in calories, and we're typically going to report that in the calories with a capital C. And chemists can also use the term joule. It's just another type of energy unit. So very similar to the difference between a pound and a kilogram. The calorie versus the kilojoules is slightly different conversion factor, but the same sort of concept. So we're gonna stick with calories just because that's typically used in medicine. So the nutritional calorie is going to be a kilocalorie, remember, which is 1,000 chemist calories. So this is how a calorimeter works. You'd have your food sample in the center there. You would have the ignition wires that would provide the start or the little spark to get that food sample burning. There's oxygen in the sample as well. We have sufficient oxygen to keep this burning. We're stirring the water around this thing, and then this little steel combustion chamber is going to heat up, and that's going to change the temperature of the water, and we can use that to calculate how much energy was given off. The more the temperature of the water changes, the more energy was given off in this process. So when we talk about burning calories, I'm gonna jump ahead a little bit here and talk about oxidation reduction reactions a little bit on this slide. We'll get into more detail on those. But we know that when we burn things, we're adding oxygen. So if we remember when we discussed some combustion reactions, we're taking a fuel, typically a hydrocarbon, and we're reacting that with oxygen. So that's a process called oxidation. We're adding oxygen to the carbon and to the hydrogen. So we're taking some hydrocarbon, reacting with oxygen, and making water, which is H2O, and CO2. Both of these are hydrogen with oxygen and carbon with oxygen. So we've oxygenated it. We've added oxygen to it. So our bodies do that same thing. So this is an example of, these are all examples of exergonic reactions. We burn calories. I want to point out that some of the foods that we eat contain different amounts of calories, or different amounts of energy. So carbohydrates, when we go to study carbohydrates, we're going to learn that carbohydrates are already partially oxidized. They are carbon-containing molecules that already have some oxygen. So the less oxygen you have, the more saturated you are with hydrogen rather than oxygen. The more oxygen you have present on the molecule, the less energy you have. Essentially, it's already in a lower energy state because it's already oxidized. So if you just have a lot of, like a strong hydrocarbon, such as in our fats, we learned that fats are big, long hydrocarbon chains. That's going to be more stored energy. So there's a lot of energy stored in that carbon to hydrogen bond, more so in that than in that carbon to oxygen bond. So things like our carbohydrates and proteins that have a lot of oxygen or nitrogen already incorporated into that molecule are going to have fewer calories per gram of the material. So in this table 5.1 is showing carbohydrates and proteins, very similar amounts of calories per gram of material, where our fats have a lot more energy. And this is why our bodies store energy as fat. This is because it requires energy to move material around. And so it wouldn't be smart to store all of your energy in something that requires a lot of mass or doesn't have a whole lot of calories per gram. So fat is a very efficient way to store and move energy. Your books provide this table, and I really don't like this table because the masses don't all add up to the same thing. So you really can't compare one food to the next and go, okay, this is the healthier food, or this is the better food to eat. But it just gives you a general breakdown of like what's in an apple. So don't try to compare between the different rows because they're not really comparable. It's literally an apples to oranges comparison. But it can kind of, if you look at one row individually, kind of give you some interesting information. For example, if you eat an apple, pretty aware that apples contain mostly carbohydrates. They're primarily carbohydrates. It's sugars and starches stored in the white part of that apple. There is a small amount of protein because proteins are the machinery that life uses to build things. So there was protein involved in the building of all that sugar and starch that's in that apple. And there are live cells in that apple, and all cells have a cell wall, and cell walls are created by fats. So we have fats involved in that apple as well. And then you can just go on down the line and check out all the different types of foods and take a look at the different ratios of carbohydrates to proteins or fats. These are kind of our macronutrients that we discuss in nutrition classes. So I want to talk about low calorie foods. This is something that's kind of interesting. I think it's really interesting. We know that most of the calories when we drink like a soft drink or something that tastes very sweet come from sugars. So you can kind of fool the body by putting something into your body that has the same structure as a sugar. It's definitely a sugar, but it's not the types of sugars that we have evolved to break down and convert into energy. So you can take substances such as dextrins and celluloses. These are molecules that we can't break down, but they are structurally very similar to the sugars that we've evolved to eat and break down. And so your tongue recognizes them as sweet. Oh, this is a sugar. I like this. Give me more of it. And so a lot of our artificial sweeteners and things along those lines uh, come from molecules like that resemble sugars. So the next thing we're going to discuss is kinetics in chemistry. So kinetics is the study of the rates of reactions. We can determine the rate of reaction by measuring the amount of product that's formed and dividing that by the time it took to form that amount of product. So this can typically be done by like purifying it and measuring the amount, or sometimes products have a different chemical absorbance spectra, like they'll absorb light differently, so we can actively measure it while the reaction is going on. There are many different ways that we can measure these processes. So in order for a chemical reaction to occur, the two molecules in the reactants must bump into each other. And they have to do that with sufficient energy and in the right orientation to get this to occur. So we'll show some graphical representations of this. And when we talk about the formation of proteins and DNA and the synthesis of these long biological molecules that are big polymers, we'll talk about how the different building blocks have to be brought into the right orientation in order for that to happen. So the two must bump into each other, and not every time that reactants bump into each other doesn't always create a product. They have to do it with enough energy and in the right orientation to fuse together to form the product. So any factor that we can do that affects the rate of collision or the way that the molecules are oriented could influence the chemical kinetics. So if we want to speed something up, we could do a few different things to speed up the molecules, perhaps, to increase the number of times they're bumping into each other. And if they bump into each other more often, then they're going to be in the right orientation more often, or they're going to hit each other in the right orientation more often just because they're hitting each other more often. So we'll show some different examples here on this next slide. So this is an example of a chemical reaction. We'll say we have the blue chemical here, and we have the red chemical here, and these are our reactants. So these are the two things that we're starting with, and they're going to bump in together and create this new thing. So we could kind of consider that to be sort of an A plus B goes to C situation. So we have that particular chemical reaction. So there are a few ways that we can speed that up. So the first way that we can speed up this chemical reaction, we want to make C faster, is we can heat this up. We can put a Bunsen burner under there. We can add energy to the reaction somehow. So our body does this all the time by coupling the breakdown of ATP to add energy, speed things up. So that's one way. So these molecules are going to start moving faster. They're going to bump into each other more often. And because they're bumping into each other more often, they might bump into each other in the right orientation more often to form more of the product. Another way that we can speed this up is we can increase the concentration of one of the reactants. So let's say that A are the blue spheres. So we'll say that A is cheap, B is kind of expensive, but we want to speed this up so we can make more C. So we'll just add more A. By adding more A, there's more A's for the B's to bump into. So there are more of the blue spheres for the red spheres to bump into. So the rate in which they bump into each other in the right orientation has gone up. So we can increase the temperature to speed up how often they bump into each other in the right orientation and speed, or we can increase the concentration of one of the reactants, the guys that we're starting with. The third way is that we can create a catalyst. A catalyst is a molecule that grabs a hold of both of the other molecules and forces them to bump into each other in the right orientation. The neat thing about catalysts is the catalyst is not consumed in this product. This guy could grab a hold of a blue sphere and a red sphere, fuse them together, and pop off and do this again and again and again. So the catalyst itself is not consumed. The enzymes in our body that are mostly proteins in our body that drive chemical reactions or biochemical reactions are catalysts. So you can have the same, we'll say phosphofructokinase is an enzyme that's involved in the processing of glucose to make energy. So one phosphofructokinase molecule could break down thousands and thousands of sugar molecules. It could be involved in the combustion of thousands and thousands of sugar molecules before it itself is finally broken down or recycled in our body. So just to review this, because this is a big thing for the test, understand what factors can we use to influence the rate of a reaction. We could increase the temperature. Increasing the temperature is going to increase the kinetic energy of the molecules. They're going to start to move faster. As a result, they're going to collide with each other more often, causing more reactions to occur. What if we want to slow a reaction down? We would try to cool it. So we would try to add something that's cold. We do this all the time in firefighting. So if there's a massive fire, we are dumping water onto this process. Water can absorb a lot of heat. So water takes energy out of the system. It slows the molecules down, and you slow down the rate of the reaction. You slow down the rate of that fire. So temperature can be used in both directions. This is also why things start to go haywire when our body temperatures aren't in the right range. So you can slow up, or you can slow down or speed up biochemical reactions by changing your internal body temperature. And that can throw off the delicate balance of our equilibrium in our system. So we're at a very delicate chemical equilibrium. And if you slow down certain processes or decrease the temperature, that could result in drastic health consequences. It's the same with speeding things up. There are other mechanisms that temperature, there are other ways that temperature influence biochemical reactions. So it can change the shape of proteins. It can change the fluidity of the cell wall. So there's other things besides speeding up and slowing down the temperature of reactions that are the rate of reactions that temperature is involved in. And it's not the only reason that it's important to maintain a fairly regular body temperature, but it is one factor. So the second thing that we can use, we mentioned, was the amount of reactants. So again, if you add more of one of the reactants, the molecules are going to have more of an opportunity to bump into one another. Now, this we're going to bring up a second point to that. The rate of reactions often slow down. Sorry, I'm trying to circle this bottom guy here. The rate of reactions often slow down as reactants are consumed to form products. And we'll discuss why that is right now. So as the reactants are consumed, you're running out of reactants. And because you have fewer reactants in the system now, they're going to bump into each other less often, and the reaction is going to begin to slow down. So here's a representation of that. So when you start a reaction, you're going to start with a lot of the reactant, and you're just starting to form products. We have a reaction start here. You've got a lot of A and B, and this is a reaction where A plus B goes to make two new guys, C plus D. We could think of like methane plus oxygen goes to make CO2 and water. When you first light the fire, you've got a lot of methane and a lot of oxygen. About halfway through the fire burning, you're going to start to have more equal amounts of reactants and products. So about halfway through, the reaction is going to start to slow down because there's less A and less B to bump into each other. Once we near completion here, we're going to really have very little A and B left. So the rate in which they're bumping into each other is going to slow down significantly, and we're going to start to see that the fire is not burning as brightly as it did before. You can see this with different things like a candle going, or when you start to run out of propane in that propane tank when you're grilling, the flame starts to get lower and lower and lower, and all of a sudden it's out. So that's where we're nearing completion. Because we're running out of reactants, there's not enough of them to bump into each other to keep this process going any longer. Earlier, we mentioned activation energy. The activation energy is the energy that's required to get this process to start. Typically, the activation energy is the energy that's involved in that chemical collision. So you have to have enough energy for these guys to collide, and they have to do it in the right orientation. A catalyst speeds up a reaction by lowering this activation energy. It does that by grabbing a hold of the two molecules and forcing them into the right orientation. So if they're already lined up just right, it doesn't take a whole lot of energy to get them to fuse together. You could think of this as like if you had two Legos. I could take those two Legos, and if I didn't line them up the right way, I could still get them to fuse together if I had a big press, or some big mechanical device where I could just put so much energy into those things that I physically fuse them together. But if I have them lined up right, I can just do that with my bare hands, right? They're a Lego. I can just line the top part up with the bottom part, clip it together. So my hands would be acting like the catalyst. They're just lining it up in such a way that it doesn't take a whole lot of energy to get the reaction to go. So the catalyst, an important point of the catalyst, is neither a product nor a reactant. Oftentimes our catalysts are either going to be like acids, bases, metal ions, and importantly in biological chemistry, enzymes are going to be our catalysts. So enzymes are our biological catalysts. Enzymes can be protein. They can also be made of RNA. We have RNA enzymes, DNA enzymes out there. So there are different types of biological molecules that can function as enzymes, but most of the time it's going to be protein. Here's a great example of one biological catalyst. It's an enzyme called catalase. So it even has a term or the root name of catalyst in it. So hydrogen peroxide is a compound. It's actually a biologically relevant compound. So our bodies produce hydrogen peroxide, and our bodies use hydrogen peroxide kind of the same way we use hydrogen peroxide. We use hydrogen peroxide to kill bacteria. So use it to sterilize things. Hydrogen peroxide is toxic at the high enough doses. Our body can make it in low doses and use it as it needs to, but it would also have to be able to break it down. So if our bodies couldn't break down hydrogen peroxide, we probably would not use it in the way that we use it today. So without this catalase enzyme, hydrogen peroxide would be a pretty potent poison to us. It would break down our lipids, oxidize our cell walls. So this particular molecule, H2O2, is a very strong oxidizing agent, and it reacts with lipids and fats, and it breaks down the bacterial cell wall. Our bodies have an enzyme called catalase to protect us from that reaction. So this catalase enzyme grabs a hold of two H2O2 molecules and forces them in such a right orientation that it pops the oxygen off of each one of those guys and forms O2, and you get 2H2O in the process. So there's an enzyme catalase, grabs a hold of two of these hydrogen peroxide molecules, fuses them together so they pop off and produce O2 and H2O as well. And that catalase enzyme in that process is not broken down. So it can repeat this process over and over and over again until it's gotten rid of any toxic levels of H2O2 in the cell. So I mentioned earlier, enzymes are biological catalysts. They're really important because we can't really do some of those other things that we could do in a flask to speed up a reaction. We can't do that in our bodies. So your body has to maintain a constant temperature. So you don't have the opportunity to we'll just heat ourselves up and get that reaction to go fast. You can't do that. The amount of the reactant present in cells is typically constant. So you don't want a big massive change in glucose flushed into your cells. You want to keep that kind of similar. So we have to use catalysts to drive our chemical kinetics in our bodies. So we use enzymes. Most enzymes are proteins, but as I mentioned earlier, you can have RNA enzymes, some DNA enzymes. They really enhance the rates of chemical reactions. Some enzymes can do this by more than a factor of 10 million. So it really speeds up the rates of chemical reactions, which is why we can respond so quickly. We can all of a sudden go from a sitting state to a running state and not run out of energy. because our body goes, oh crap, we need to burn glucose now and make more energy. We need to do this really, really fast. So it has these enzymes that can burn glucose a lot faster than you could burn glucose if you were setting it on fire on a table. So if you had a pile of sugar, you could catch that sugar on fire with a match. It will burn. Um, but our enzymes do that at a very rapid rate, so we can produce enough energy to keep us going. Enzymes are very highly specific, so they bind or they speed up reactions by immobilizing the reactants or grabbing a hold of the reactants at an active site. And I have a diagram for that on the next slide here, so you can see a visual representation of that. And they'll grab a hold of these in an active site, which is like a little pocket in the enzymes. You think of an enzyme as a little globular ball, and it's got a little pocket that it can grab a hold of the reactants and orients them in the right fashion, and the reaction occurs. Enzymes are very specific, and if there's a mutation in the gene that codes for that enzyme, if that mutation is in the active site, that can be a devastating disease-causing mutation. So sometimes it doesn't really affect the active site to the point where um, you'll see a decrease in specificity or a decrease in function of that enzyme, but sometimes it does. So a lot of enzyme-based diseases exist out there, and there's a lot of research going into DNA editing. So will we be able to, in future generations, edit the DNA in an embryo so that your child would not pick up on this genetic disease? So these are things that, 10, 15, 20, 100 years down the road, we might have that technology. And this is the graphical representation of what we're doing, or a pictorial representation of what an enzyme is doing. You kind of have these little pockets here. So I've got my enzyme, and this would be part of my active site that binds to this substrate or the reactant. Substrate's what we call things that proteins bind to. So they grab a hold of a substrate. So substrate is just a fancy word for reactant. So it's grabbing a hold of reactant A, and then it grabs a hold of reactant B, and it's putting them in these little binding pockets. And now they're in the right orientation that they're going to fuse together and float off into space now. And then the enzyme is refreshed. It's ready to rock and roll again. So it's not burned up in this process. There are a lot of examples of enzyme-based diseases, but these are just a couple of examples. So albinism, the enzyme is tyrosinase. So this presents, again, absence of pigment in the skin, hair, eyes. Gout, this involves an enzyme that breaks down ribose sugars. This can create renal problems, joint problems, high levels of uric acid in the bloodstream. Lactose intolerance, this is very common, and this can kind of occur as you age too, where the lining in your gut can stop producing an enzyme lactase, or not produce as much of the enzyme lactase. And lactase is a sugar that is present in particularly high concentrations in dairy products. So that sugar, if it's not broken down in your stomach, it can be broken down in your gut by bacteria that create gas. So if it's not broken down by lactase in your stomach and small intestine, if it gets into the bowel, it can create diarrhea, bloating, flatulence, all sorts of problems for people with lactose intolerance. So those are several examples. Then we're going to move on to discuss the different types of chemical reactions. We're not going to discuss all types of chemical reactions, but we're going to cover some of the big guys. So we can classify reactions in these three different ways. We could talk about synthesis reactions. These are reactions when smaller molecules are combined to create larger molecules. Typically, these are going to be endogonic processes or endothermic processes. This is going to require energy. Not always, though. There are some synthesis reactions that do occur spontaneously. These typically involve man-made compounds like chemical polymer or monomers that we use in polymer synthesis. So these polymerization reactions can be spontaneous, but most synthesis reactions require energy because you're taking smaller things and putting them together. If you think about that, that's like ordering something. So you're taking smaller things that are lower energy molecules, and you're building these bigger, larger energy molecules. So most of these processes require energy. And that's synthesis reactions. Decomposition reactions would be the opposite. So you're taking large molecules and you're breaking them apart. So typically these are going to have these are going to be exogonic or exothermic reactions. These reactions will typically only have one reactant. You're going to be starting with one big reactant, like the reactant is A, and that gets broken down into multiple parts. So it could be like A goes to B plus C. An exchange reaction, we call it sometimes a displacement reaction. Those involve both kind of like a synthesis and decomposition. So it's typically your A plus B goes to C plus D, where somebody gets traded out. Partners are getting switched here. So that would be an exchange reaction. We'll show some examples of those. In a synthesis reaction, you have two things that are getting converted into a bigger thing. So you have A plus B get converted into some new molecule that is a fusion of A and B. In decomposition, it's the opposite. You have the fused product and it's being broken down. In an exchange reaction, some partner is getting switched out. So you have the fused A and B, and it's trading the B for the C. So the B pops off, the C gets added. Or you're going to have like a double displacement. So you can have A and B plus C and D, and they trade partners. So the last thing we're going to discuss before we take a little break here, we're going to have two parts to chapter 5, are reversible reactions. So reversible reactions are reactions that can go in either direction. So you can your reactants can form the products, but then the products can start to break down back into the reactants. So these typically happen in biological processes, and we can kind of think of the formation of taking CO2 and water to make glucose, and then going back to CO2 and water is a reversible biochemical reaction. So you can have photosynthesis would be going in one direction, and glycolysis, the breakdown of sugar, would be going in the other direction. This would be an example of a reversible reaction. So chemical bonds can be formed with input of energy, and they could also be broken. So you could go back in the other direction. So it's very important to understand that biological processes, keeping a balance of the different chemical components in your body is often very important. So enzymes are often have evolved to go in both directions. So a lot of biochemical reactions are going to be reversible reactions. Where if you start to if you want a balance of A and B and the fusion product here, and so if you start to build up too much of this, you can start to go back in the other direction to make it more of your starting materials. So just to reiterate that, so you could have a forward reaction that occurs when A and B form this fusion product, AB or AB. When you have a sufficient amount of that fusion product that builds up, the reverse reaction could start to occur. You start to have the breakdown of that to reform the starting materials. There will come a point where both the forward and reverse reactions are happening at the same rate. When this happens, it appears as if the concentrations of both the reactants and the products are not changing anymore. They've reached what we call a chemical equilibrium. This does not mean that there are an equal amount of reactants and products. That is very important. There could be a lot more product, there could be a lot more reactant. It means the reaction reaches a point at which the rates of the forward and reverse reaction are the same. Doesn't mean the concentrations of the products and reactants are the same. It means the rates at which they're being formed and broken down are the same. The reactions that we have that are very highly exothermic are what we call, for all practical and purposes, considered irreversible reactions. So an example of that would be rejoining combusted molecules. If you take methane, which is CH4, and react that with oxygen to make CO2 and water, it would take so much energy to form methane out of CO2 and water again that it's just practically impossible. So we call that an irreversible reaction. In order to get methane again, we're going to have to turn that CO2 and water back into sugar, and some body is going to have to do a whole bunch of chemical reactions to that sugar to turn it back into a fat, which is a hydrocarbon, and then we're going to have to break that hydrocarbon down into methane gas again. So there's a whole bunch of steps. It's not really a reversible process. You can't just go from methane to CO2 and then straight back to methane. It's an irreversible reaction once that gets broken down and burnt down.